Is it recording?
Hi, I'm Jessica. I'm adult number 1.
Hi, I'm Joanne, adult number 2.
Chris, number 3.
Okay. And my name is Allie, and today I'm going to be talking to you about Dolly the sheep. On July 5th in 1996, a medical marvel was born. This medical marvel was named Dolly, and in this talk I will explain to you where, how, and why Dolly was created. Next slide. To begin, Dolly was formed from the mammary gland of a Fen Dorset sheep and a separate egg cell from a Scottish black-faced sheep, who was her surrogate mother. Here is a picture of Dolly and her surrogate mother, the black-faced sheep. Next slide. Dolly had a profound impact on the scientific community because she was the first successfully cloned mammal that was developed from an adult cell. Her conception from her surrogate mother led to a lot of questions, but ultimately scientists were able to tell she was a successful clone simply because she was born with a white face and body, whereas her mother was a black-faced sheep. Now I will explain to you in greater detail why exactly scientists look to cloning mammals. Next slide, please. Now, at this point, you may be wondering exactly why a scientist would want to clone a sheep or any mammal in general. That is a very good question, and one that I certainly had while researching information for this topic. Dolly was created at the Roslin Institute in the United Kingdom. Before her creation, the main purpose was to develop a successful method for successfully cloning livestock, meaning that if the series of experimentations was successful, the Institute wouldn't have to use as many animals for future research. Another important reason scientists were researching cloning was because they wanted to learn more about cellular reactions during fetal development when made from a skin or brain cell. They were also researching the medicinal properties of mammal milk. In an interview with Sir Ian Wilmot, he was asked about the tissue used to clone Dolly, to which he responded, "It was mammary tissue. Our collaborators remember the project to make sheep that produce their milk proteins needed to treat human disease." Here's a picture of Dolly with the lead scientist, Sir Ian Wilmot. Next slide. While the Institute's main purpose for cloning Dolly was to research the medicinal properties of milk, years later, that is no longer were the main purposes of cloning. In modern times, cloning has the biggest impact on stem cell research. Shinya Yamanaka, a stem cell biologist, Nobel Prize winner, and university professor, claimed that the success of Dolly's cloning motivated him to begin researching stem cells that had been derived from adult cells. Shinya began experimenting on mice and was able to create iPS cells, which has reduced the very need for embryonic stem cells, which has been a very large source of controversy. There has been brief discussions about cloning humans, but cloning expert Professor Robin Lavelle Badge stated that given how inefficient and definitely unsafe it is, he would be crazy to try it, and there has been no good justification for doing it. Next slide. Now that we have discussed why exactly scientists were looking to clone Dolly, I will go into more detail about the process the team of scientists went through to create her. When asked to briefly explain the process of cloning, Ian Wilmot said, "You need 2 cells. You need an egg from which you remove the genetic information, the chromosomes, and you need a nuclear donor, which will provide the nucleus that controls development." So in the case of Dolly, this was a mammary cell, which was in culture, which was used and proved to be able to control development. Unfortunately, it was a difficult process for the team of scientists to be able to successfully clone Dolly. Before she was born, the scientists had made 277 previous attempts. As they first began cloning Dolly, they altered the growth medium and injected the udder cell into an unfertilized egg cell, which had the nucleus removed. They were able to fuse the cell by using electrical pulses. After the team of scientists successfully managed to fuse the nucleus, they began culturing it for a period that lasted about 6 to 7 days, in order to see if the cell would divide and develop normally. Luckily, the results were positive, and they were able to successfully implant her into the surrogate mother. The reason Dolly is so special is because out of 277 cell fusions, only 29 embryos were able to develop and were implanted into 13 different surrogate mothers. Dolly was the only pregnancy that was able to survive and was successfully born. Next slide. Dolly lived the majority of her life at the Roslin Institute in the United Kingdom. It was within the Roslin Institute that Dolly was able to mate and breed normally, and even successfully had her very own set of offspring. And there's a picture of the offspring and Dolly. Next slide. Now I'm going to explain more in detail the scientific results of Dolly's birth and life. Since Dolly was the first big breakthrough in cloning mammals, the scientists discovered some unfortunate details concerning her DNA. Since Dolly was a clone of a 6-year-old sheep, her DNA was that of a 6-year-old sheep, even though she had just been born, meaning that even though she was still young, her telomeres were much shorter than normal sheep her age. Telomeres are very important because they cap the end of the DNA molecules, ultimately protecting the molecules from damage. It is normal that telomeres begin to lessen in size as aging continues, but since Dolly's DNA was 6 years old at birth, her life was very short-lived. Sadly, on February 14, 2003, Dolly was euthanized due to health conditions at only 6 years of age. The decision was ultimately made because she had been suffering from severe arthritis in her hind leg joints that, even though were treated, did not show any signs of improvement. Then the farm workers noticed Dolly began to develop a cough. When the scientists took her in for scanning, they noticed she had a large tumor in her lung, which is not uncommon among sheep who have been raised indoors. Next slide. After Dolly's death, the Roslin Institute transported her body to the National Museum of Scotland, where she still remains one of the most popular exhibits within the museum. Next slide. So what have we learned about Dolly, the first cloned sheep? We learned that Dolly played an important role in science because she was the first mammal that was successfully cloned from an adult cell. And the DNA from that single cell was powerful enough to create another adult sheep. We also went over why Dolly specifically was so special. That is because out of 277 attempts of cloning, Dolly was the only sheep to be successfully born. Dolly's birth was transformative to the scientific community as a whole, and I hope you all enjoyed learning about her as much as I did. Thank you.
How do you stop it?